Hello, my name is Darwin, and I am a graduate student at UCSF studying under the co-mentorships of Dr. Hideo Okada and Joseph Costello. And today I'll be presenting my work on tumor-wide RNA splicing aberrations that generate public immunogenic neoantigens. So a very common problem with cell-based immunotherapy is that it is very limited in cancers with low somatic mutational burden and high intratumoral heterogeneity. And these tumors include glioblastomas, low-grade gliomas, medulloblastomas, and many other cancer types as well. These immunotherapies actually work very well in cancer types with high mutational burden, such as melanomas. But in those with many subclones and a divergence of evolutionary trees, you have these subpockets where neoantigens either don't exist or express at very low levels. So discovering new tumor-wide neoantigens from other sources could actually potentially expand the repertoire of immunotherapeutic targets and address this challenge of intratumoral heterogeneity. Our project focuses on using RNA as a potential source for generating these neoantigens. Studies have shown that RNA splicing events can actually generate immunogenic targets, but no study has actually investigated the clonality of these targets themselves. Many have discovered neoantigens that have worked and have not. But our study actually hypothesizes and looks for public and intratumorally conserved immunogenic neoantigens that can be generated by clonal tumor-specific alternative splicing events in gliomas and other cancer types. So we generated a pipeline that we called SNP, standing for Spatial Splice Neoantigen Identifier Pipeline. And this pipeline identified a total of 789 public neo-junction candidates in GBM and LGG cases using TCGA and GTEx as the main source of data. We've done this for other cancer types as well, and we've actually used this pipeline to identify not just neojunctions that are found in TCGA, but those that were found spatially conserved throughout the entire tumor across multiple sites within the same sample. And we have done this for many other tumor types, but specifically for GBMs and low-grade gliomas, we actually have a database that is specifically stored for 10 spatially and maximally distant samples from the same tumor, and we were able to identify neoantigens and neojunctions that were spatially conserved, meaning that they were found in multiple, if not all, of the sites within that same tumor. We have used mass spec and RNA sequencing to determine that these neojunctions are indeed real. And we've incorporated many other algorithms into our pipeline, such as HLA and McFlurry, to determine that these peptides are indeed correctly cut and presented by HLA. We then were able to identify TCRs that specifically targeted these neojunction-derived neoantigens and prove that they are indeed immunogenic. And we were able to put them into CD8-positive T cells and other T cell modules as well, and determine that these actually elicit a cytotoxic response against the tumor cells and also cells that present the neoantigen artificially. And as such, we have identified tumor-wide alternative splicing neojunctions that generate clonal immunogenic neoantigens for T-cell-based therapies. And neojunction-derived neoantigens are actually shown from our pipeline to be endogenously processed and presented to elicit a T-cell-mediated response. So what we're hoping to do with this study is to open the door to a new interpretation of neoantigens and a new class of them where intratumoral heterogeneity is taken into account. And hopefully this will be brought into a more high throughput pipeline and can be applied for more cancers beyond GBMs and LGGs as well. And thank you so much, Ars, for all of your support for this project.